What if, just by looking at the blueprints of life, we actually change it? We're going to dive into a breakthrough so big, it actually creates a brand new paradox, right at the very heart of biology. So let's really get into this. Because look, this isn't just about making science faster. It's way deeper. We're asking if the very act of predicting something perfectly changes what it means to discover it in the first place. Does the AI's gaze transform the molecules it sees, or does it really just transform us? All right, so to really get what a huge deal this is, we've got to rewind the clock. We need to talk about one of the biggest, most frustrating puzzles in all of biology, the protein folding problem. Okay, picture this. The year is 1969. A guy named Cyrus Leventhal throws down this mind-bending paradox. He calculated that a single protein, just one, could theoretically fold itself into 10 to the 300th power different shapes. I mean, that number is so big, it makes the number of atoms in the entire universe look tiny. And yet, in our bodies, these things snap into the exact right shape in microseconds. Like, how? For half a century, that question was a total mystery. So to tackle this, scientists set up this big competition, right? It's called CASP. And for decades, I mean decades, progress was, well, slow. A good score was maybe a 40. Then, boom, 2018. DeepMind's AlphaFold shows up, and the score just leaps. But wait, 2 years later, AlphaFold-2 doesn't just win the competition, it basically ends it. A score of 92.4? That's not just good, that's pretty much perfect. We're talking atomic-level accuracy. The race was over, just done. And you can't overstate how disruptive this was. I mean, think about the old way. You had these brilliant but super slow methods like X-ray crystallography. We're talking months, sometimes years, and a ton of money just for one single protein structure. Now, we're in a world where you can get a super accurate prediction in minutes. Minutes! The game completely changed. Time, scale, cost, all of it. And this, of course, led to just an absolute explosion of data. In just a couple of years, the AlphaFold database went public. We're talking over 200 million protein structure predictions, available to anyone with an internet connection. The analogy is perfect. It was like going from having a couple of old hand-drawn maps of your town to suddenly having Google Earth for the entire biological universe. And this new map is already helping us solve huge, real-world problems. For example, it helped us finally figure out the structure of the nuclear pore complex. That's basically the bouncer at the door of our cell's nucleus. It decoded the SARS-CoV-2 spike protein in what felt like a blink of an eye, which massively accelerated vaccine development. And hey, for all you in veterinary science, it's predicting the shape of proteins in cow udders to help fight mastitis. But here's the real kicker. It cracked a stubborn little protein called APHID1503, something that had stumped the best labs in the world for 10 years. So that's it, right? Problem solved. High fives all around. Well, not so fast. This is where our story gets really interesting. What happens when our main way of seeing biology isn't through a microscope anymore, but through an algorithm? Is that perfect digital blueprint really the same as the messy, chaotic, living thing? Think about it. The scientific method itself is getting flipped on its head. For centuries, the process was clear. Observe something in the lab, cook up a hypothesis, and then test it. But now, for a lot of researchers, the first step is to ask the AI. You predict first, and then you go to the lab to see if the computer was right. That's a massive, fundamental shift in how science gets done. Now, here's the really clever part. AlphaFold is actually smart enough to know its own limits. For every single prediction, it gives a confidence score. It's called a PLDT score. You can think of it kind of like a weather forecast. If it's green, the AI is saying, yep, I'm pretty sure about this part. But if it's yellow or orange, that's the AI basically raising its hand and going, I'm not so sure here. This bit is kind of fuzzy. And guess what? Those fuzzy parts, that's often where the magic happens. And this gets to the heart of it. AlphaFold gives you this picture-perfect snapshot of the final destination, but it tells you next to nothing about the journey. Take the tau protein, which is linked to Alzheimer's disease. AlphaFold can show you its nice, neatly folded state, but it totally misses the messy, dynamic, tangled-up process that actually causes the disease. It's like it can show you exactly where a car is parked, but it has no idea about the chaotic traffic jam it went through to get there. So this all leads to a really deep, almost philosophical question. We're essentially creating these digital twins of biological molecules. We can poke them and probe them and test them on a computer before we ever step into a lab. But What does it mean to make an authentic discovery anymore? Are we actually studying life, or are we just studying a really, really good simulation of it? And this whole dilemma, this paradox, it actually has a surprising parallel in a completely different field. I'm talking about quantum physics. It's almost like biology now has its very own quantum uncertainty principle. Okay, quick physics refresher. Back in the 1920s, Werner Heisenberg figured out something wild about the quantum world. You simply can't know everything about a particle all at once. The more you pin down its exact location, the fuzzier your knowledge of its momentum becomes, and the other way around too. It's a fundamental trade-off baked into the universe. And what's crazy is, AlphaFold seems to have stumbled into the exact same kind of trade-off. So here is the quantum parallel in biology. The more precisely we know a protein's final, perfectly folded structure, let's call that its position, the less we seem to know about the dynamic, chaotic process it took to get there, its momentum. By handing us that perfect final picture, the AI is unintentionally hiding the very thing we wanted to know in the first place. How does it solve Levinthal's paradox? We get the destination, but the map of the journey just vanishes. And look, this isn't some rare little problem. We're talking about a third of all our proteins having these fuzzy, low-confidence zones. And they're not mistakes. Often they are the most important, most functional parts of the protein. The classic example is p53, the famous guardian of the genome. It's a tumor suppressor, and its power comes from its flexible, disordered regions that let it grab onto DNA and stop cancer. So it turns out, uncertainty isn't a bug, it's a feature. It's where the action is. This leads to a really dangerous new kind of human bias. Let's call it predictive myopia, right? When you have this unbelievably powerful tool that gives you what looks like a perfect answer, it's so tempting to just accept it, to believe it 100%. We risk getting tunnel vision, where we only look at the solved, high-confidence parts and completely ignore those fuzzy, uncertain regions, which might be exactly where the next Nobel Prize-winning discovery is hiding. So, does this mean human scientists are out of a job? No way, not even close. This new world of prediction doesn't make humans obsolete. It just demands that we evolve. It calls for a whole new kind of scientist. The biologist of tomorrow needs a totally new toolkit. Yeah, you'll need to know your way around a computer, for sure. But even more important is learning to think like a Bayesian. Always questioning, always weighing the AI's confidence against the evidence. You'll have to work with people from totally different fields. And most importantly, you have to bring the stuff no algorithm can. Ethical judgment, human intuition, and that spark of pure creativity. So stop thinking about it as AI versus humans. The future is an AI and human partnership. Here's how it works. The AI proposes. It generates this incredible first draft, a hypothesis. Then the human validates. You become the creative director, the chief skeptic, the explorer. Your job is to design the smart experiments to test that hypothesis. And even more importantly, to go exploring in those very same fuzzy areas the AI told you it didn't understand. And this powerful new partnership forces us to face some really heavy questions. I mean, if we can predict your risk of getting a disease just from your genome, what happens to your privacy? If we can use this to design new forms of life, who gets to write the rules? These aren't sci-fi questions for the future. They are on our doorstep right now. So maybe, in the end, we just need to look at this whole thing differently. Maybe the uncertainty that AlphaFold points out, those fuzzy, low-confidence spots, maybe that's not a flaw in the system. Maybe it's the most important feature. It's like the AI is handing us a treasure map with big red circles around the unexplored territories saying, go look here, this is where the adventure is. Look, this technology is a tool. It's arguably the most powerful tool biology has ever had, but it's still a tool. It's a curiosity amplifier, not a curiosity replacement. AlphaFold can predict the folds, but it's up to you, the next generation of scientists, doctors, and thinkers, to predict what comes next and to build the future of life. So what's the bottom line? It's this. We have to lean into these incredible new powers. We have to predict boldly. But at the same time, we have to question everything, even more deeply than before. Because the real breakthroughs aren't going to come from the answers the AI gives us. They're going to come from the brand new questions it finally lets us ask.